Hello everyone, my name is Christian Perea and I am in Professor Stiver's class, and today I will be presenting a video on mitosis. I hope you enjoy. Now everyone come around and gather over here. I've got some of a rap right now that you're about to hear. On my mind, I got this thing called mitosis, but if they aren't lined up, it'll look like a lot like scoliosis. Of course, scoliosis, the spine is not straight, and of course with mitosis, the stages, of course, are pretty straight. It's pretty plain and simple. You've got about 6 different stages. Their orders are interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. The way I remember it is IPMAC, which is broken down as investigating pets may attack the cat. In interphase, chromosomes replicate in preparation for mitosis. Then we go to prophase, which have nuclear membrane breaks, and then they break down. Sister chromatids, which are replicated chromosomes, and then they condense. And then the spindle form. Going to metaphase, the sister chromatids line up at the center of the cell. The third step of mitosis is anaphase, when the sister chromatid pairs are pulled apart by spindle fibers. One full set of chromosomes goes to one side of the cell, and another goes to the other. The fourth step of mitosis is telophase, which the chromosomes begin to uncoil as the nuclear membrane is resembled around them. Last but not least, the cytokinesis has the cytoplasm and organelles duplicate and are divided into approximately equal parts. The cells split into two. Right now, I'm about to break it down real nice and easy. Now here's a photo of what I think is interphase. I'm just kidding, I wrote it down. So here's interphase. Of course, you see the nucleolus, and I didn't write on top, but you see the centrioles and the centrosomes. And you see the chromatin inside, which of course are going to be the chromosomes later on. And then the nuclear envelope, which are surrounded around the cell. So now I'm going to break it down and go to the other side, which is prophase. So here's a photo of prophase. You see here, stuff has, of course, broken down. And you see the nuclear envelope, you see the chromosomes forming the chromatins, of course, the nucleolus, and then the centrosomes and the centrioles on top. So now I'm going to go to the other side, which is the next step of the stage, which is metaphase. And here you see the spindle fibers, which I had said earlier, they are forming. And then you still see the centrioles and the centrosomes. And then you see the chromosomes are lining up. So I'm running out of smooth lines now, so I'll just come out and say it. Here is the next step, which is anaphase. So anaphase, you see the chromosomes are pulling apart. You see the centrosomes and the centrioles, of course, on the sides. And the spindle fibers, which are right here. I'm going to go to my next stage in mitosis, which is telophase. Telophase is right here. So you see the cytoplasm, of course, it's pulling apart. You see the chromosomes right here. You see the nuclear envelope. And then, sorry, my finger's in the way. You see the centrosomes and the centrioles on this side right here. Last but not least, the last phase was the non-rhyming ending, cytokinesis. So you see there's two new daughter cells, of course. You see the centrosomes on the side, on centrioles on each side, the nuclear envelope and the chromatids. So it's basically another interphase, and this is where mitosis restarts again. I'm talking about mitosis. No. Not meiosis, but mitosis. Unfortunately, since we are talking about this subject, there is something that can go wrong. Babies are a miracle. Let's get that straight. No matter what, it's a blessing to have in the world. But in the case of an extra chromosome, that 21st chromosome sometimes has an extra. This goes to show you that nothing in the world is perfect. But Down syndrome is the name. Trisomy 21 to be scientific. Now here's a baby that doesn't have chromosomes, and here's a baby that doesn't have an extra chromosome, and here's a baby that has Down syndrome, which has an extra chromosome. So here's a photo. Sorry about that. And here's a photo of a baby that has Down syndrome on this side, and then the one that doesn't have Down syndrome. So you see the facial features are a little bit different, and the eyes are a little bit different. Of course, you see them over time um, on the streets or whatever like that. I know it seems I'm going out of order, but I'm really not. The mitosis definition is a process in which the nucleus of the parent cell duplicates. Mitosis has a pretty straightforward purpose. It is enabling existing cells to generate new genetically identical cells. There's a chapter in the book saying most cells are not immortal, which means, of course, mitosis generates replacements. This makes it possible for organisms to grow and replace cells that die. A fact about chromatin is that it is not available until mitosis. Now, here's some different examples of mitosis. A couple definitions needed to be explained are spindle fibers, chromosomes, and centrosome. Spindle fibers are basically what cuts in the half of the cell. So you saw those in the earlier photos that I showed you. Chromosomes are packaged thread-like structures, so the ones that kind of look like this, and then of course all over. Centrosome is an organelle in the cell that is basically with the spindle fibers that form, and of course you saw them on the side. And thank you for listening. Now this is the ending of my mitosis video. Thank you.